Hello, everybody. I'm Professor Rodrigo Rezende from University of Araraquara, from São Paulo State, Brazil. And today we will have a nice presentation from Professor Dr. Lorenzo Moroni. He is professor at the Maastricht University, Merlin Institute, Department of Complex Tissue Regeneration, the Netherlands. The title of his presentation will be Biofabrication for Regenerative Medicine Applications from Acellular Instructive Scaffolds to Bioprinted Tissues. Professor Moroni studied biomedical engineer at Polytechnical University of Milano in Italy and nanoscale sciences at the Chalmers Technical University, Sweden. He received his PhD cum laude in 2006 at the University of Twente on 3D scaffolds for osteochondral regeneration. Dr. Moroni worked at Johns Hopkins University as a postdoctoral fellow in the ELISIF lab, focusing on hydrogels and stem cells. Since 2014, he works at Maastricht University, where he is a founding member of the Merlin Institute for Technology-Inspired Regenerative Medicine. In 2016, he became full professor in biofabrication for regenerative medicine. His research group interests aim at developing biofabrication technologies to generate libraries of 3D scaffolds able to control cell fate with applications span from skeletal to vascular, neural, and organ regeneration. He received several awards during his academic career. So thanks a lot, Professor Moroni, for accepting participating in our 3DBB event from Araraquara, Brazil. It's really a great honor to count on you, on your presence here this day. Have a nice presentation.
Thank you, Rodrigo. Thank you for the kind introduction and for the invitation to share with you some of our results and some of our perspectives around the topic of biofabrication technologies applied to the regeneration of tissues in our body. It's a pity I cannot be there physically with all of you. Of course, the timing is not the easiest to travel. Nonetheless, I hope that I will be able to interact and show you some exciting and interesting results. Now, the field of biofabrication stems for many of the technologies that are into this new field of science from the additive manufacturing field, where originally now, almost 30 years ago, we started to use additive manufacturing technologies as a way of creating prototypes. And at that time, the technologies were also called rapid prototyping, with the idea of recreating a technical drawing in real life as a sort of demonstrative sample before actually moving to real industrial manufacturing. This set of technologies, in the meantime, have become more and more actual production tools and production technologies. First, in order to create different kind of tooling or molds, and then really moving to actual digital manufacturing. And now more and more being considered as personal fabrication. Again, in non-biomedical field, we can think of interior design or art objects which are really personalized thanks to the use of additive manufacturing technologies. It's a growing field with a high impact, with an expected value of several hundreds of billions of dollars to reach in the 2025 or around 2025. In the context of biomedical applications, and more specifically of tissue engineering and regenerative medicine applications, biofabrication technologies have been defined by a number of active scientist in the field, following essentially two main pillars: that of bioprinting, where we direct, especially the arrangement of cells, materials, and or growth factors or biological factors more in generally, or that of bioassembly, where actually by again using automated assembly, cellular building blocks can be placed together to recreate a macroscopic tissue from elements that are typically more featured in the micron size domain. In both cases, then the tissue equivalent or the tissue construct is also left to mature in vitro for a number of days or weeks, and then is implanted or is used as an in vitro model to understand better the kind of pathological or pathophysiological biological events that are behind either a tissue disease or actually the tissue regeneration that we would like to aim for. Now, within these two domains, actually, we also conceive a third possibility within biofabrication, which is that of the use of additive manufacturing or other kind of automated manufacturing technologies for the fabrication of acellular constructs, acellular scaffolds, which are considered biofabrication only when the design actually results in either smart hierarchical or surface properties able to steer cell fate, and in particular stem cell fate. And so within this general introduction, additive manufacturing for sure are the pillar still of bioprinting approaches. And you see here also electrospinning as being a technology that is continuously used towards sort of automated deposition of fibers. Again, both technologies can also and have also been used for the fabrication of such acellular instructive scaffolds, whereas more bottom-up approaches are conceived to create those kind of biological Lego-like blocks, if you wish, from which we can then build the full Lego house, if you wish, and therefore in a parallel the full biological tissues. Within the additive manufacturing context, and again more specifically the techniques that have been used for the fabrication of scaffolds or the fabrication of biological constructs in our able to be used in our body, there are a number of different advantages and disadvantages that I will not go through. Again, here we have reviewed them with a number of other members of the community in a paper of a couple of years ago in Trends in Biotechnology, where we also looked at the fabrication capacity in terms of speed and resolution of each of these technologies. And that's what you see here in the graphic, where electrospinning is probably on the low side in terms of speed of fabrication, but that has a very high resolution, whereas other kind of technologies such as SLS, FDM, or bioplotting are on the higher side in terms of speed of fabrication, but also are more limited in terms of the resolution of the minimum features that can be produced with such technologies. And so, stemming actually from the experience of our lab, actually, I just would like to show you some examples where either in terms of acellular construct or going more and more towards complexity, biological complexity, where then we do use bioprinting technologies. We hope we have actually contributed to part of the regenerative medicine growth of the last few years. Now, if you think about a simple way you can create a hierarchical acellular scaffold is that actually of changing the pore size or the pore shape as you build such kind of 3D printed scaffold. So layer by layer, you can choose at a predefined layer to then change the size of the pores as here depicted by the scanning electron microscopy on the top right of the slide. And we have done that trying to approximate as much as we could biological principles. And if you think about skeletal tissues, typically an osteochondral defect, where you have both articular cartilage as well as the subchondral underneath bone, you would have gradients of many different biological factors, among which gradients of oxygen, where typically you would have low oxygen closer to the cartilage side and higher oxygen closer to the bony side of such osteochondral scaffold. So we created such a sort of pore size gradient axially through the scaffold fabrication methodology. We then cultured adult bone marrow derived mesenchymal stem cells, or stromal cells, more properly said, for up to 35 days, either in basic or in mineralization media, or actually in chondrogenic media, to see again the possible effect of both osteogenic and chondrogenic differentiation of such cells. And actually, if you see here the level of alkaline phosphatase activity of these cells, we can actually see that in mineralization media in the gradient scaffold, here represented by the blue contour and the blue bar in the graph, we had a significantly higher amount of such a protein compared to the non-gradient scaffolds made of homogeneous pore size in the lowest or highest range comprised into the gradient construct. If you would then cut the gradient construct into the different pore size regions, we would see that such an increase of ALP activity was directly proportional with an increase of the pore size, being highest around a pore size of 900 to 1,100 micrometers, so in the order of sub-millimeter scale. Vice versa, if we would now look at the same sort of study, but then in chondrogenic media, we would assist to an increase of GAG, or glycosaminoglycans, production, both in this case for gradients and non-gradient scaffolds with the smallest pore size. And again, if we look at the gradient scaffolds and we cut it into the different segmental units, we would assist at such an increase being inversely proportional to the pore size. So highest with the lowest pore size used in the scaffold fabrication. So again, in the order of 500 microns. This actually confirming a little bit what was our original hypothesis, that probably with lowest pore size, we have a tendency of having a lower oxygen and therefore higher matrix production for cartilage tissue, and in higher pore size, we would have higher oxygen concentration, higher nutrient availability, therefore more predisposition for osteogenic differentiation. To further confirm that, we also looked at a specific protein which is directly related to the actually the lack of oxygen, which is called the epoxy reducible factor. And indeed, we saw that both in the variety of 1 alpha as well as 2 alpha, the protein was much higher present in the lowest pore size compared to the other pore size of the gradient scaffolds. So again, further confirming this relation between predisposition of cells into differentiating towards the osteochondral tissue versus the possible level of hypoxia in these pores, and therefore the possible local level of oxygen concentration. Now, if you consider of possible gradients in other tissues in our body, essentially this of osteochondral tissue is just one example. Other kind of systems could be, for example, ligaments or tendons. And typically, this is not only structural gradients, but also physiochemical and biological gradients. If you think again about a tendon tissue, you see here, for example, how different kind of proteins and different kind of genes vary from the tendon itself to the insertion point into the bone to the bony side itself as well. A number of attempts actually have tried in the field to create scaffolds that could replicate some of these features, but all of them, and also the example that I showed from our lab with 3D printing, have been discrete gradients, so gradients which have been made only with a change every certain amount of layers or every certain size of each region. In fact, actually, if we think about such kind of gradients in our biological tissues, these are much more continuously present in our body. And so, with that idea in mind, actually, we also developed more recently a new additive manufacturing technique where essentially we combine blending with classical extrusion-based deposition. So in this case, we can change the composition of two materials while we print in the same layer, and we don't therefore not being limited by changing the material composition every certain amount of layers. And you see here on the left an example with just a food dye, so that's where the scaffold changes from the more whitish color of the original polymer we used into a more pinkish color where the highest concentration of the food dye is present, and the right side actually a real biological case where in this case we have added inorganic compounds containing actually antibiotics, and that's why you see the brownish color, which is not the polymer being burned, but it's actually the color of the inorganic compounds that we have included into the blending system while printing. While doing that, we have actually produced a number of different nanocomposites, either changing the level of antibiotics that we would be using in such inorganic fillers, or more considering bone or osteogenic differentiation, the amount of reduced graphene oxide, which has been shown to have a strong effect on osteoinductive induction of MSC differentiation, as well as non-hydroxyapatite or microhydroxyapatite, which are commonly used in the context of bone tissue regeneration. And we see that by changing different kind of composites in their chemistry or in the amounts of the fillers into the polymer, we actually could modulate extensively the mechanical properties of such kind of composites, spanning to the blank modulus of the polymer used here, a copolymer of polyethylene oxide terephthalate and polybutylene terephthalate, to up to 600 megapascal in the best case scenario, with a 45% inclusion of micro-sized hydroxyapatite, which starts to get closer to actually subchondral bone properties. Now, is this continuous gradient really any better than discrete gradients? From a mechanical point of view, this seems to be the case. What you see here also in the video is that while the discrete gradients here on the bottom left video tend to fail progressively at each different sub-region of the scaffold, in the continuous gradient, actually we can compress the scaffold homogeneously and continuously throughout its volume before it fails, and this is also represented into the stress strain curves, where you can see that the scaffolds actually have a much higher strained break and a much higher stressed break compared to the discrete gradients. We also use different kind of characterization tools, from backscattering electron microscopy to Raman spectroscopy and micro-CT, to really evaluate whether the gradients are continuously changing throughout the scaffolds, and have them tested these scaffolds both in vitro as well as in vivo. Here you see the fabrication of a critical size defect, a non-union representing a non-union fracture in a rabbit model, where we actually chose two kind of gradients, either mimicking the continuity on the bony stumps of the non-union fracture, therefore having there the highest amount of hydroxyapatite, or simulating a sort of callus region with the highest amount of non-hydroxyapatite in the center of the construct. What we saw in vitro is that, again, these kind of scaffolds induce a high amount of alkaline phosphatase activity, which is an early osteogenic marker, but also the increase of more mature proteins such as osteocalcin, which is further expressed typically at later time points. So in the span course of 5 weeks of culture. From a mineralization point of view, which is of course important in terms of bone tissue regeneration, we also saw that with a 45% of hydroxyapatite included into the scaffolds, we had not only a higher amount of mineralization in mineralization media, which is sort to be expected, but also interestingly in basic media. So again, showing the possible osteoinductive capacity of this kind of scaffolds. We have also further increased the level of control of in space and time of cell adhesion of this kind of scaffolds with the integration of a atmospheric plasma torch, which has allowed us to really change also the kind of hydrophilicity and hydrophobicity of the scaffolds, therefore creating local areas of adhesion and versus local areas of non-adhesion of within the scaffolds. And this, you could imagine that it could be used to, for example, seed the two different kind of cell populations, which therefore can recreate a more complex tissue interface in the regenerative processes. Again, here think about the osteochondral interface, where cells typically would be more rounded in the cartilage side and more spread on the osteogenic side. Now, changing also this kind of parameters during manufacturing and the kind of polymer composition has also allowed us to study more rationally the kind of physical chemical properties of these polymers in during printing. And so we have created a sort of printability window where now we can predict by measuring the complex viscosity of a number of different molten polymers and understand actually what are the time span and the temperature span conditions for which such kind of polymers can be printed. And that's work from a PhD student called Andrea Calore. Now, what actually do we need more in terms of instructive characteristics? If we look, for example, at one of the most performing synthetic material in terms of osteoinductive character, that of ceramics, we have seen that from literature in the field, that actually the microtopography of such kind of ceramics can play a drastic difference in terms of changing the material, the same sort of chemistry of the same ceramic from non to pro-osteoinductive. This also has been somewhat studied also in polymers by the group of Matt Dalby a number of years back, where actually they showed the creation of nanotopography with specific size and actually also specific disorder resulted into increase of osteogenic markers in vitro. So inspired by these two works, actually what we have done with Honglin Chen, a former PhD student in the group, now a group leader back in China, was actually to spin a relatively bioinert ceramic material such as zirconia, and then during the sintering phase, change actually selectively either the roughness or the crystal size of the formed nanofiber scaffolds. Now, the first thing actually that was particularly interesting was that these electrospun mats in fact seem to be quite flexible, which for a ceramic material is quite unique as a property. And in fact, we also tried to burn the material to see if this was due to some sort of polymer remnants into the scaffold, but that was not the case. You can see that really, whereas at the single fiber level, the stiffness of the material is the same as the bulk stiffness of a disc of zirconia, at the mat level, at the scaffold level, the mesh actually is really much more flexible. And this also has been coupled to fatigue tests, where these kind of scaffolds have been able to sustain cyclic loading up to more than 4,000 cycles. We then again cultured adult bone marrow-derived stem cells on these scaffolds, and indeed, interestingly, we found that already in basic media there had been a increase in gene expression of a set of both early as well as late osteogenic gene markers, especially for those scaffolds that have been sintered with at a lower speed, so in an oven at 5 Celsius per minute, which actually were the scaffolds with the highest nano roughness on their surface. Again, this was not only at the gene level, but also at the protein level. We assisted at a higher ALP activity, again, in this case, only in the osteogenic media. But again, interestingly, as a ultimate test, this also was associated to an increase in the mineralization potential of these scaffolds, again, in this case, only in mineralization media. Now, changing here from 3D printed scaffolds to electrospun scaffolds, one of the main limitation of such scaffolds is also that typically cells have a difficult life to penetrate through a full three-dimensional construct, so that oftentimes electrospun scaffolds are considered semi-3D or still a sort of 2.5D system because of this lack of cell penetration inside thick electrospun meshes. So to solve actually this possible problem, actually we adopted a sort of simple method where we would spin on top of a pre-stretched film, which then would be left to unstretch, and by doing so create a sort of buckling effect on the fibers, on the electrospun fibers, which would result into this kind of waved pattern on the fibers. So indeed, if we do this now, and so in a cartoon with real polymers, we did assist to an increase on the waviness of such kind of fibers, not only on single fibers, but also if you would spin for long enough, you see up to 45 minutes also at the mesoscale on the actual fiber mesh itself. This actually was interesting because it also allowed us to change, by changing also the polymer characteristic, the amplitude of these kind of waves, and to create some sort of undulated structures that are also resembling tissues in our body. So for example, here on the left upper side, you see the undulation of collagen fibers in the heart, whereas if you go more towards the lower side, you see examples of tendon ligaments, and in the last case of the ciliary body in our eyes. The buckled scaffolds actually did their job. Not only they had also a modulation in the mechanical properties, but also, and more importantly, resulted in the migration of and population of cells through more than 1 centimeter in thickness of this kind of constructs. But interestingly, what we also found was that the waviness of this kind of structures resulted in the selective expression of TGF-beta, which is a growth factor that is also common into tissue morphogenesis and especially connective tissue regeneration. So we are now also testing this kind of waved scaffolds into a ligament model to see if the waviness pattern could also influence the regeneration of connective tissue such as tendons and ligaments. Now, of course, when we think about more complex tissues in our body, it's not only about the resident cells of those tissues, but also the associated networks. And in many cases, actually, our tissues and organs can't live if there is not an adequate vascular and an adequate innervation to the tissue. So that's actually where when we start to look at these more complex, multicellular and multi-tissue kind of constructs, that bioprinting or bioassembly can become more useful. And here you see actually a number of different approaches in terms of bioassembly. what I did, as I mentioned at the beginning, different kind of blocks could be created a priori and then joined together to create a more macroscopic tissue. This is beautiful work from the group of Shoji Takeuchi and the group of Alike Demostheni, among others, that we have reviewed also recently in the field. What actually we have done here in a project that hopefully is with a good name for the Brazilian audience is actually the creation of a sort of injectable smart paste, and there is music in the between of the video. But essentially, what you see here in our cartoon video is that the idea was to create a certain kind of micro objects, and then by engineering the properties of these micro objects, we could use them to create a hybrid injectable system with cells, which activity could be modulated by the properties of the micro objects themselves. And we do this in collaboration with the group of Roman Tuchel-Müller, also at Moroni Institute, who is an expert of micro and nanofabrication technologies. So what we have done was first with technical photopolymer materials such as SU8, which are not necessarily something we want to have in our body, but as a proof of concept showing actually that cells, depending on the geometry and the size of these objects, actually could really manipulate and put together also rectangular beams into macroscopic sized objects. And then if you would pile on top of each other these different kind of mesoscale objects, you could really create up to centimeter-like scaled construct. And here you see a kind of toroidal construct representing a sort of tracheal-like tube, so for our respiratory conduit. We then moved to biomedical grade materials such as PLLA, again looking at both a different kind of shape and size of these objects and understanding more fundamentally from a biophysical point of view how these objects actually influence the circularity, therefore the level of compaction and aggregation of cells into such a hybrid injectable system. And started to also mix different kind of cells. You see again mesenchymal stromal cells and endothelial cells to really start creating vascularized injectable units. Alternatively, what we could do is with bioprinting really deposit in different layers or within the same layer with different kind of fiber properties, such as, for example, shell core fibers, different kind of cells. Now, in the case of bioprinting, there are a number of technologies that have been developed. The three most used are laser-induced forward transfer, where droplets of these cell-laden hydrogels are being deposited on a stage, or that of inkjet printing or robotic dispensing, where again, either by droplets or by continuous filaments, we can deposit cell-laden hydrogel constructs or actually cell spheroids without the use of hydrogels. More interestingly, recently, also levitation principle has been developed by the group of Utka Demirci and Vladimir Mironov in the biofabrication field, where actually they showed it was possible to really position in space different kind of cells by exploiting either acoustic or magnetic levitation. Within our department, actually, we are busy with different kind of applications in the context of bioprinting, stemming out from the osteochondral defect applications. That's where we started in the presentation. More and more towards building complexity, and that's where our efforts on cardiac islets and kidney and thyroid are currently focused on. We do that also with different kind of researchers in the institute at different departments, such as Art Van Apeldoorn for islet of Langerhans and Stefan Giesebrecht for thyroid constructs. Now, I'd like to focus just to give you an example of the complexity on kidney bioprinting, which is also a one of the efforts in bioprinting that has most recently attracted attention in the field. And this is actually work under the lead of a young faculty member of the department, Carlos Mota. Now, in the case of kidney, of course, if you see the level of proteins and different kind of structural features and functionality that one would have to recreate, this could really fit the purpose of using more complex biofabrication technologies such as bioprinting. So that's what we have started, and considering the fact that also the kidney is populated by more than 20 different kind of specialized cell types, we also thought to start from pluripotent stem cells and use them to recreate our bioprinted concept. And at the beginning, we had to optimize protocols for differentiation of pluripotent stem cells into the different kind of differentiated kidney cells. That's what a very skilled postdoc in the lab, Abhishek Harichandan, did in the past 4 years. So first, not only being able to maintain cells undifferentiated, but then really more specifically looking at different level of differentiation of such IPS into progenitor and as well as already differentiated kidney cell types. And here I'm not going into details on the different markers, but essentially we were able really to replicate different kind of level of differentiation of different kidney cells into the from the starting IPS derived cells. We then also further went down into either uterative bud or metanephric mesenchyme progenitor cells. And you can see here the typical feature of these uterative bud cells is the formation of this kind of branching structures. We then started to look at bioprinting, but again here we started to face really the challenge of how delicate are IPS-derived cultures to be deposited, where in principle, considering different kind of commercially available gels, only high stem, which is a modification of hyaluronic acid with PEG and gelatin, resulted in some level of acceptable viability, despite again the fact that here you see still a high number of cell death, which is something we have to still improve upon. Another way to further increase the vitality of the cells actually is to look at methods where a lower concentration of gel could be used. Again, here you see that by using simply alginate and reducing the concentration from 4% to down 2% or even 1% alginate with 1% of matrigel, sorry, it was possible to really obtain now much more satisfactory viability of these organoids after bioprinting. We also started then, obviously alginate is not sufficient alone, so we also started to look at the cellularized extracellular matrix as a way to create a biological cocktail that could be added to alginate. And here you see that after 5 days, cells are getting a much better viability state. Not only that, but they could also express specific markers, again, of differentiation towards the kidney, specialized lineages, both in 2D as well as when encapsulated in 3D into this kind of ECM-derived hydrogels. Now I'm towards the end of the presentation, where I would like also to show just a sneak preview of some of the work that also to another young faculty member of the department, Matt Baker, we are doing, and that is on the creation of new bioinks for bioprinting. This has been work done by another skilled postdoc in the lab, Oy, who's recently changed towards industry after finishing her postdoc. And in principle, what Matt and Hui have done was again starting from alginate as a building block that of changing the backbone to the reader, not only cross-linking through calcium cations, but also through UV sources by adding actually a tyrosine or bromine linker. This actually allowed us to create much thicker constructs and also to spatially control two different kind of cell types. Here again, green and red are cells actually that have been stained with a cell tracker, with two different cell tracker in the same construct, again showing the possibility of pattern in different cell types within the same construct. Cells actually were alive over a week of culture at different kind of level of stiffness of the constructs that we were able to create. So I hope that I have showed you that we can today create different kind of simple tissues for implant up to 3D in vitro models where pieces of organs or lobes of organs could be conceived. Probably to really think of full organ bioprinting, it will be a much longer endeavor where maybe one day in 20, 30 years from now, we will be able to assist to such kind of dream. What are the steps that we would be in need of thinking in this direction? Which kind of cells would be used, whether we would be able to conserve the functionality of the targeted organ, and whether which kind of technology and scaffold we would have to print and use, and in principle whether we would be able to recreate the same multi-scale structure and the same sort of multi-cellular composition of such kind of complex structures in our body, as well as whether we would be able to integrate also vascular, neural, and lymphatic networks in them. Finally, our tissues are not only static materials, but in fact, if we consider them as biological materials, they are very much dynamic. And that's also what in the group of we are now focusing at in the creation of bioinks that can be dynamic in their origin, and by exploiting supramolecular chemistry, we are now able to create bioinks that not only create again vascular-like structures, but also can self-heal when broken, and depending on the level of dynamicity on the gel, actually are able to support a more rounded versus a more spread cell morphology. With this, I'd like to conclude. Actually, I was a bit longer than originally planned. I'm sorry about that. I would like to, of course, introduce the faces of what I call since a few years the superhero in the lab. Superhero because it's a highly interdisciplinary group of people spanning from biologists to chemists, going through a number of different engineers. You've seen the faces, you see the names. Also, thanks go to the funding bodies, without which support it would be impossible to do the research. And finally, I would like to thank you for listening and invite you next year here in Maastricht, where we will be organizing the World Conference of the Thermi Society. So if we will be allowed again to travel, I hope to welcome you and warmly welcome you there in Maastricht next year. Thank you.
Hello, Lorenzo. Thank you so much for your nice presentation. It's really a great honor and happiness for us here in Brazil. I met you maybe 8 years ago in Biofabrication in Manchester, and I think that we talked sometimes after, and as a result, we have the pleasure to have you here this day. And so I hope, as you said in the beginning, that we can meet each other physically after this pandemic situation, right? And I hope that you can come to Brazil to visit us.
Yeah, that would be lovely.
And so thank you so much again, and we see each other soon. Bye bye.
Right. Thank you, Rodrigo.